Good morning, guys. It has been a while since we have been in class. And so I'm going to walk you through the next section of notes and also do a little bit of practice with you to kind of get you rolling. So as we start this, I want to make sure that you have the documents that you need to make the most out of this little mini lecture. And it really is a mini lecture. So let's make sure you need your notes packet that looks like this. So pause and pull this up, go find this. We are in this section right here. If these are not filled in, it's probably because you haven't done a previous, this was a pre-recorded lecture for you, pushed out mid last week. Dihybrid crosses we did mostly in class. So we are now to this point. We are starting topic 5.4, which is all about differences from sort of the classic, what Mendel thought was the way that traits were inherited. His idea was called complete dominance, which means if you have one dominant allele, you're going to express the dominant trait. And although that is true for what he saw in pea plants in particular, we know that there are other instances that kind of fall outside of that. So we're going to look at what are called non-Mendelian patterns of inheritance. So you need this notes packet. And then you're also going to need this. So if you were in class, the kind of early dismissal day, which I believe was last Wednesday, the sub actually handed you a packet full of papers to kind of take home and carry you through. Those were A-day students. So if you're an A-day student, you have this already printed out. The sub gave it to you. If you're a B-day student, unfortunately, we didn't get back to school on Thursday for you to get these packets. And so these are linked on the calendar. So just backtrack to the calendar real quick and snag these. You'll want to print them out. For this one, we are only doing the first 2 pages. So if you're printing this at home, you're only printing page 1 and 2. Do not keep going on and on and on. Just print the first 2 pages. So once you have your notes and you have this document ready to go, then come back to this video and let's do the lecture. All right, so like I said, we're now getting ready to sort of move away from or deviate from how Mendel thought alleles express themselves. So remember, Mendel thought that if you inherited just one dominant allele, you would show the dominant phenotype, meaning if you are homozygous dominant, you would show the dominant phenotype. But if you were a heterozygote, you would also show the dominant phenotype. And then, of course, if you're recessive, you're going to show a different recessive phenotype. So really, Mendel thought there's really 2 possible phenotypes. You're either the dominant or you're the recessive. Well, what we have come to know and understand now is that although, like I said, although that is correct for what he observed in nature, there are other patterns of inheritance that are different from what Mendel initially thought. And so the first one that we're going to talk about in this section of notes is what is called incomplete dominance. So I'll give you a second to kind of peek at this picture. So check this out. You have a red flower that is crossed with a white flower, and it's showing you in the parent generation, the red flower is homozygous dominant red. The white flower is homozygous recessive. And so if I really did the cross between these 2 parents, all of their offspring would come out being heterozygous. Now, if this were Mendel, we would say that all of these heterozygote babies would be red, just like that dominant parent phenotype shows. But what we find here in this case is that the heterozygous is actually a brand new color. So we see the emergence of a third possible phenotype. You might be red, you might be white, or you might be pink. And so incomplete dominance is when the heterozygote, right? The heterozygote shows a brand new phenotype. This is our third possible phenotype now that could enter into play. So in this case, the third new color is pink. And you can see down here that same example. If you are homozygous dominant, you are red. If you are homozygous recessive, you are white. If you are a heterozygote, you are pink. So think for a second, why do you think this new phenotype is pink? Because it's kind of a blend of the previous two colors. Okay, so in incomplete dominance, we say that no one allele is truly dominant. And so when we get the heterozygote phenotype, we get a blend of the homozygouses, right? So we get red, sort of just like you're mixing paint. Red blends with white and gives us this pink color. And so in complete dominance, nobody is truly, truly dominant. And the heterozygous offspring gives us a new phenotype. In this case, the phenotype, in incomplete dominance, the phenotype is a blend of the other two. Now, I want you to look at, here is another same example, red flower, white flower crossed together to give us a pink flower. But I want you to check out how the genotypes are written. So up here, this is pretty standard. This is what we're used to. Big R, little r, big R, big R, little r, little r. Here is even what we're used to. Big A, big A, big A, little a. But this is also another way of showing alleles. And so what you see here, look at this genotype. You see a big C, and that C stands for color. And so that lets us know that the trait that we're tracking in this cross is the color of the flower. And then the actual genotype, what we're used to looking at, is writing up here almost like an exponent. It's a superscript letter. And so for the color trait for the red flower, we are big R, big R. The color trait for the white flower, we are WW. The color trait for the heterozygous is one of each, one R, one W. Now, some of you might be going, wait. This is new. I don't like this. What happened to the same letter? Let's use a big one and a little one. That is fine, but you still need to be able to recognize what it's representing. Because remember, guys, these are just models for inheritance, right? And so the models might look like what you're used to, but the models might also look like this little exponent kind of genotype. It means the same thing. It's just a different way of kind of illustrating this particular genotype and this cross. So we'll see both. We'll see both kinds of examples, and that's really in an attempt to get you comfortable with seeing both for the AP test. Okay, so incomplete dominance, I'm recapping. Incomplete dominance is when no allele is truly dominant over the other, and they kind of mix together in the heterozygote. Now let's shift for a second to the practice sheet that you have and actually hop down, because incomplete dominance is the second set of questions. So hop down to number 6 on this practice sheet. You can pause the video at any time. I'm going to keep going, but if I'm talking too fast, just pause it and get yourself situated and then unpause and move forward. Right? Okay, so we're going to try 2 incomplete dominance practice questions together really quickly. The first question set, number 6, this is getting us comfortable with recognizing genotypes and phenotypes. So it says here, in snapdragons, which are the flowers we just saw, in snapdragons, flower color is controlled by incomplete dominance. The 2 alleles are red, big R, and white, little r. The heterozygous genotype is expressed as pink. So really quickly, if they had left out this part of the sentence is controlled by incomplete dominance, they didn't tell me that, I could still understand this to be incomplete dominance just by reading the characteristics. 2 alleles, 1 red, 1 white, and the heterozygous gives us a new color that is a blend of the other two. Right? Gives us pink. So that could cue my brain to say this is incomplete dominance if the question hadn't already said it. So A says, what is the phenotype? Phenotype is the physical characteristic. What is the phenotype of a plant that is big R, big R? What is the phenotype of a plant that is heterozygous, big R, little r? And what is the phenotype of a plant that is little r, homozygous, recessive? So take a second and answer those. Now let's move on to number 7. Remember, if I'm going too fast, pause the video. Number 7, a pink flower is crossed with a white flowering plant. What is the probability of producing pink flowers in their next generation? Really, if I had finished out that, it'd be better off said in the next generation. So a pink flowering plant, think about what that genotype would be. Crossed with a white flowering plant, think about what that genotype would be. Do the cross right here to the side, and then answer the question. Probability of producing a pink flowered plant. When you're ready to move forward to the answer slide, unpause the video. Here's the answer. So check yourself up here. What is the phenotype of a plant that is big R, big R? It is red. Now, I also wrote out here to the side just so you could get perspective. You do not need this written down, but I wanted you to see if this genotype were written with the C's and the superscript, this is what it would look like. What is the phenotype of a plant with genotype big R, little r? It's pink. Here's what that genotype could have looked like. Instead of big R, little r, it could have looked like this. And then what is the phenotype of this last one? It is white, and its genotype could have looked like this. That got smudged. That looks funny, but that's a little r. Okay, so then check yourself down here, number 7. A pink flowering plant crossed with a white flowering plant. Here's your cross. 50% of their offspring could also be pink. All right, so let's go back into our notes. All right, so pause on your worksheet. Let's go back into our notes. Let's talk about codominance for a second. So codominance is a little bit different. This is both alleles actually are equally dominant. So in incomplete, we saw that no allele was truly dominant. Codominance, we see that both alleles are actually truly dominant. So check out this little fish picture right here. You have a red fish crossed with a blue fish, and look at the heterozygote is both red and blue. Both of those pigments are expressing at the same time because they are co-dominant pigments. Same thing down here, a white cow mixed with a red cow produces what's called a roan cow, which is both pigments produced at the same time on that cow. So the heterozygous now takes on both phenotypes at the same time. And again, I would encourage you, check out the funny genotypes, right? So it's the same thing. They're written the same way that we saw in the incomplete dominance, where we see those little they're kind of hard to see in this, but we see those little superscript letters. This is the heterozygote. And if we could see those tiny little superscripts, we would see them to be probably big W, little w, or a W and an R, white and a red. So let's go to the practice questions for codominance. Okay, so number 2 here in the codominant section talks about the fish. It says in certain fish, blue scales and red scales are codominant. When a fish has the hybrid genotype, hybrid is another way of saying a heterozygous, right? Pure breeding is a homozygous. Hybrid is one of each, one dominant and one recessive, quote, recessive allele. The hybrid is a patchwork. Okay, so this is asking you to use letter B, and this time it's asking you for the genotype. So what is the genotype for a blue fish? What is the genotype for a red fish? What is the genotype for a patchwork fish? So because it asks us to, using the letter B, you could add those right here. What are their genotypes? If you want to go like super test yourself, you can use the funny genotypes, the superscript, kind of the ones that look like exponents. And then number 3, what happens if you breed a patchwork fish? So patchwork, that's our heterozygote, with a fish that has blue scales, which up here, blue scales. So if we breed those two together, do the cross, and then answer the questions. What's the probability of having fish with red scales? What's the probability of having patchwork scales? So try that out. And when you're ready, unpause the video and we'll go to the answer slide. Here we have answers. So here's the genotype. Using just the letters B like they suggested, blue fish are going to be big B, big B. Red fish are going to be little b, little b. Patchwork fish will be the heterozygote, big B, little b. You could have kind of tested yourself and done the funny genotypes, the ones that look like exponents, and there they are. Number 3, what happens if you breed a patchwork with a blue scale? Here's that cross. Patchwork is our heterozygote. There's our blue scale. So I do the cross and then I answer the questions. What is the probability of having fish with red scales? 0. We're not going to get any 0, or we're not going to get any red scales. What's the probability of having fish with patchwork? 50%. These two down here. Okay. So that sort of wraps up our codominant and incomplete dominant lecture and a little bit of practice. Now, what you're going to do is finish out the rest of those codominant and incomplete dominant questions. So remember, this is different than what Mendel said. And the big difference for both of these is that the heterozygous genotype is going to show a whole brand new phenotype. If it's incomplete dominance, the phenotype is a blend, like you're mixing paints. If it's codominance, the new phenotype is sort of both at the same time. Speckled, spotted, patchwork, dotted. The roan cow is a good example as well. So finish out this practice sheet and you'll be good for incomplete and codominance.